unique and identical.
But you can have two different types.
So we have asexual reproduction, which is where the offspring are clones, so they're genetically identical to the parents. And then we have sexual reproduction, where you have two parents that are genetically non-identical, so they're unique, and the offspring is a mixture of those two parents.
Alright.
So now we're going to have some genetic diversity. What do you think that might mean? What's genetic diversity? Any guesses? What?
The genes are different.
Yeah. What could be some benefits of genetic diversity?
Might get favorable traits from either of the parents.
Excellent.
Rather than just from one parent. Makes less susceptible to disease.
Oh my gosh. Did you read the notes beforehand, or are you just reading my mind?
No.
Sorry. Okay, so as we increase genetic diversity in a population by undergoing sexual reproduction rather than asexual reproduction, we can make the members of the population less susceptible to disease and more responsive to changes in the environment. So if we had animals that were all genetically identical and then a disease came along that they were susceptible to, it'd wipe out the entire population, and that would not be ideal for the population as a whole. So by being genetically diverse and non-identical, the whole population is more fit and more responsive to changes. So this will make the whole group more capable of surviving and reproducing. So this is a lot of this chapter will be centered around ways that we're increasing genetic diversity in a way that still makes functional organisms. So we don't want them to be so genetically diverse that they don't function anymore. Because if we just mix up all the DNA completely, then we're going to have organisms that aren't going to behave properly. They're not going to be able to do like the essential roles that a cell has to carry out. So we have to increase genetic diversity in a way that still makes the cells functional, but makes them more responsive to their environment.
Unlike insects.
Well, insects do have some genetic diversity too, but we'll get into that later. Okay. Oh, incest. I thought you said insects. Oh, yeah, incest. Incest. That is a very different. We actually won't touch too much on incest in this class.
Is that why they say if the two people in the same family make a child, that the child comes out
deformed and stuff because they got too much alike?
Yeah, I'm actually not positive why incest results in more genetic defects. I guess what? Yeah, I think it's that a lot of families will have. We'll get into this more in another chapter. They'll have what are called like recessive traits for genetic diseases. What?
You said it last time.
You sure?
Well, we'll get this one. We'll get into another chapter. I feel like I covered the things I said we'll get into. Maybe not. Maybe I should have a to-do list of things that I need to cover. We'll get into that in another chapter. I don't want to waste your time now. But again, so we're going to be talking a lot about DNA and where DNA is moving around during meiosis. Just wanted to refresh everyone on what DNA is doing. So DNA is this instruction manual that allows us to make proteins. So this little schematic is called the central dogma of biology. We'll get into more what RNA is later. But it's important to know that DNA is used to encode information to make proteins. And so we've been talking a little bit about replication in mitosis. This is where we're copying our DNA. So that's this little loop here. But again, the DNA is used to encode for proteins, and the different proteins that the DNA encodes for is going to give each cell and each organism different traits and different types of behavior. So when we talk about increasing genetic diversity, we're going to be changing the form of this DNA, which is like the instruction manual for the cells. So we're going to be giving the new offspring kind of new instruction manuals. So they'll be able to make new types of proteins or new mixes of proteins that are going to give them different traits. So our DNA is packaged, again, obviously it's not obviously, it's in the nucleus, which we're all well aware of at this point. And so we have our chromosomes. With our chromosomes, we have these stretches of DNA called genes, and each gene is going to encode for a specific protein. So in the chromosomes, we have little sections that are going to encode for specific proteins, and different chromosomes and different organisms are going to have different genes that will encode for different proteins that gives them different behavior, different traits. So by changing our DNA, we can change the way that cells are behaving and responding to their environment. So we have 23 pairs of chromosomes in our cells. And so we're going to talk a little bit more later about these X and Y chromosomes. But other than that, they all come in pairs that are what we call homologous, which is like similar, nearly identical. And so when they combine, when we have two different organisms that are going to combine cells during sexual reproduction, like we said earlier, we're going to have to reduce the number of chromosomes that we have. Because if we had cells that have the full set of chromosomes, each had 23 pairs, and they combined, then we'd have 46 pairs. And then the offspring of those organisms would have 92 pairs, and it would just, expand like that. And that would not be productive. That would not be functional because we'd have too many chromosomes, and it would just increase with each generation. So instead, what we do before sexual reproduction is we have to half the number of chromosomes that we have. So then we combine, then they just have 23 pairs in the new organism. So we already answered this question, but this was on a test. What would you guys put before we get into it? A little like what's that? How do cells from two different organisms combine to make a new organism? They reduce the amount of chromosomes that they have. So that's going to be a theme in today's lecture. Yeah. We'll be focusing on those today. So the X and the Y chromosome. Oh, sorry. Do you have a question?
Yeah. What makes the decision which chromosomes to discard and which ones to keep?
We'll get into that. Yeah, it's not so much a decision as a sorting process, but if it's not clear in a few slides, let me know and we'll dig into it a little bit more. So we talked earlier about how there's these homologous pairs. So again, homologous means approximately the same size, the same genes. So they're encoding for similar things. So one particular gene could be like a gene for eye color. It's actually a little more complicated than that. But in the simplified version of looking at genes, we could say there's like a gene for eye color. So each organism could have a gene for eye color, and that would be on a specific chromosome. And even though different organisms will have different chromosomes, they'll have the same genes that they're encoding for. So each chromosome on a certain location will have a gene for eye color.
Aren't there more than one gene for all different types?
Yeah. It's usually, as biology is always more complex than it seems on the surface. So in reality, the genes that encode for eye color are spread out over many genes. And it's kind of a mixture of many different factors. But for now, we can think of it as just that there's one stretch of DNA on one specific chromosome that would encode for eye color. And then in the offspring, when we have a mixture of the genetic material between the mother and the father, we'll have a new mixture that's going to contribute to what the eye color will be. And we'll get a little bit more into the rules that govern that mixture in another chapter. But for now, it's important to realize that each chromosome has stretches of DNA called genes that encode for specific traits in the cell through encoding for RNA. And one of those could be like eye color or like hair color or things like that. There's a special pair of chromosomes called the sex chromosomes where we have either two X chromosomes here. So these are still homologous because they're about the same size and have the same genes. Or we have the XY chromosomes, which are for males. So males actually have fewer number of pairs of homologous chromosomes. They only have 22 homologous chromosomes, and they have their X and Y chromosomes, which are not homologous because they're not identical. So we've been talking mainly so far in this semester about somatic cells, or what we call diploid cells. So these have, in our body, these would be 23 pairs of chromosomes in somatic cells. So again, we can't combine two somatic cells to make a new organism because then it'll have too much DNA. So to make that happen, to be able to sexually reproduce, we produce gamete cells. So gamete cells have half the amount of DNA that somatic cells have. And those are called haploid cells. So now when we combine two haploid cells, we end up with a full set of DNA. We go from 23 chromosomes in one cell and 23 in another, and when they combine, we have 23 pairs and that's the full set of chromosomes that most humans have. So we can talk about it, for humans, we can say 23 pairs and 23 chromosomes, but to be a little more like general, broadly speaking, we can say haploid cells have N chromosomes, and diploid cells have 2 N chromosomes, where N is just like the number of pairs that that species has. Because every species, or many species, have different numbers of chromosomes.
Different haploid cells have the same chromosomes, or they have a bunch of like different.
Yeah. So let me. Oh, there goes all my stuff. I always forget which one have animations. Okay, so the way sexual reproduction is going to work is, in the process of making haploid cells, we go from having 2 N chromosomes, and we're going to produce 4 haploid cells through meiosis, which we'll get into the details of this later. And so the male and the female are both going to create haploid cells, and they're going to combine during fertilization to make a zygote. And a zygote is now a genetically unique organism with 2 N chromosomes. So would this be a gamete or a somatic cell? Somatic cell. So this is now the first cell of a new organism. So now this cell is going to have to grow through what process? Meiosis or mitosis? Mitosis, to make a whole new organism. Which one?
Blowing the color.
Oh, I thought you meant the top one.
No.
Yeah, I like.
Why is what? Good question. So in meiosis, we're increasing our genetic diversity, and in fertilization, we combine genetic material from 2 organisms, and now we have one cell that's genetically unique. But now we want this cell to grow into a new organism. But all of the cells in this organism have to be genetically identical, and they all have to have 2 N chromosomes.
And 2 N, the N stands for 23 because 23 times 2 is 42.
Yeah. So maybe we'll do, humans, N equals 23, because we have 23 pairs of chromosomes. And then, so mitosis produces cells that have 2 N chromosomes. And meiosis produces cells that have N chromosomes. So if this zygote underwent meiosis, it would just produce haploid cells, which wouldn't be very useful for the organism growing and performing all of its functions.
Which one would be 23?
Well, so, yeah, meiosis would be 23, yeah. So mitosis makes 23 pairs, or 46.
So the N represents the chromosomes that I think?
That's the number of pairs of chromosomes that we have. Yeah.
Are either of these considered asexual reproduction?
No, but asexual reproduction is like mitosis.
Okay, so it is asexual.
Yeah. But in sexual reproduction, we have to undergo meiosis to allow different organisms to combine. So, is everyone clear on the definition of the terms fertilization and zygote? So during fertilization, we have 2 haploid cells that are combining to form a zygote, which is the first diploid cell of a new organism. What? Is this better? Clear definitions? Yeah, this chapter is going to have a lot of graphic slides, but I'm sure we can all get through it because we're all very mature adults. Or we can act like mature adults for at least an hour and 15 minutes. I'm definitely not a mature
We're not
adult.
done in 5 minutes. So.
Or 5 minutes. We can do at least 5 minutes.
It's color.
What?
It's colorful.
Yeah, great. I'm glad you guys are all, and I have different colored markers too that I can use throughout the lecture too. So just lots of color.
Great.
What? Okay. Meiosis, again, we're producing 4 haploid cells. Mitosis, we're producing 2 diploid cells. So we've covered mitosis already. And the point of undergoing meiosis is so that we can undergo fertilization and make a zygote. And so the zygote is going to be the diploid cell that then grows into a new organism. So we all, at some point, we're just one single cell, just one zygote. It's pretty crazy to think at some point we were just one single cell that resulted from just 2 haploid cells combining. And now we're full-grown human beings. Isn't that crazy?
Oh, yeah.
So diploid cells come from haploid cells?
Yeah, diploid cells come from two haploid cells combining. Yeah, so maybe I should write that as well. Well, I think we'll get into that a little bit. It'll come up, I think, a few more times. But I think this illustration is nice. So we have, these are two haploid cells, each with N chromosomes, and then they combine to make a zygote with two N chromosomes, which then goes through mitosis and then undergoes this whole cycle again. So again, another cycle. There's so many cycles in biology. This is a cycle of how we can undergo sexual reproduction throughout multiple generations. And it never changes the amount of chromosomes that the organism has, like the full-grown organism has. So if we didn't undergo meiosis and we didn't have haploid cells, then the amount of DNA would just increase with each generation, and then after thousands of generations, we would have thousands of pairs of chromosomes, and that would not fit the nucleus anymore, and it would not be efficient. So this is like a more efficient way for the cell to be using its space and resources. And by being more efficient, it makes it more likely to survive. So we're going to watch a quick video to give you some more context, and we'll dig into the details. But again, the goal is to make haploid cells, and this whole process is happening in really two phases, meiosis 1 and meiosis 2. But notice there's only one interphase. So meiosis is really similar to mitosis in a lot of the steps. An interphase in meiosis and mitosis is almost identical. Why do you think that might be?
Because you have to grow.
Right, but why don't we have two S phases? Because we have two meiosis phases. Why would we not have two S phases?
We don't need to copy the DNA twice. There's 4.
Right, so we don't need to copy the DNA. What do you mean by that?
I mean, we copy the DNA and then you can keep dividing it however you want. Well, I don't know. Do we divide it twice in the same phase?
No, maybe. Do you want to add on to that?
There's 4 haploid cells, right?
Yeah.
Is that way we have to do twice?
Yeah, so let's. Oh no, I used blue again. I'm not really utilizing my color capacity. So after S phase in mitosis, how many chromosomes do we have? Usually we have 2N, but after S phase, how many will we have? 4N.
You should only need to copy it once because you only need one blueprint.
Because each of them are going to have 1 N.
Right.
So now we have 4.
Yeah, and then so I think we'll touch on that a little bit. And then we have mitosis, and we have 2N and 2N. So we went through S phase, and we ended up with 4N chromosomes, and they split during mitosis and cytokinesis to make 2 genetically identical cells, each with 2N chromosomes. In meiosis, S phase produces 4N chromosomes, and then we're going to make 4 haploid cells. So if we underwent S phase again, then we'd have too much DNA. We have too many chromosomes. So we only go through S phase once because we only need to copy the DNA once to make 4 haploid cells.
So if you ask this question, do we have to say that it's going to be like the division of the chromosomes from the number of how many diploids to how many haploids?
Sorry, what do you mean?
Are we going to have to know the number of how many it's going to divide, like how many diploids, it's going to be 2 diploids to?
Yeah, you should know that mitosis is going to make 2 diploid cells, and meiosis will make 4 haploid cells. But
Okay.
yeah, we'll get into that a lot more as we go. But I think it's important to realize that interphase only has to happen once in meiosis, even though there's meiosis 1 and meiosis 2 where we are dividing the cells multiple times.
In your picture up here, is that both meiosis 1 and 2 happening?
Yeah. So, yeah. So why don't I make that more clear? That would be good. Thank you for. So. So in meiosis 1, we end up with two cells of 2 N chromosomes, just like in mitosis. In meiosis 2, we split into 4 cells of N chromosomes each. So 4 haploid cells. So let's watch the movie, and we'll get kind of a little more context, and we'll dig into the details of how all of these steps happen. We're producing somatic cells with 2 N chromosomes. In meiosis, we're producing 4 haploid cells called gametes, and it's happening in 2 stages. We have meiosis 1 and meiosis 2. At the end of meiosis 1, we're producing 2 diploid cells. And then at the end of meiosis 2, we take those 2 diploid cells and we end up with 4 haploid cells. And in comparison to mitosis, we just end up with 2 diploid cells. So we talked a little bit about, in the video about this idea of crossing over. What was that?
So they have the chromosomes, and then like a little part of one of them, which is the little part of the other one, there's more genetic diversity.
Great. Where is that happening up here? Does anyone see? Metaphase 1. Yeah. So it's happening. You see it kind of first in metaphase 1, but it's technically happening in prophase 1. But it's kind of hard to see the smaller graphic. But in prophase 1, you have this exchange of genetic material between different non-identical chromosomes. And so this is another way to increase our genetic diversity. Should we take a break? Everyone looks really sleepy. Okay, let's take a quick 5-minute break. I know you're all excited for Easter break. I can't anticipate them. Okay, meiosis 1. No, yeah, I'm glad you asked good questions. I'm not upset, but at some point it becomes a discussion, not a lecture, which is great. Meiosis 1 is the first half of meiosis. This is very similar to mitosis in that we're creating diploid cells, but it's also different because they're not genetically identical. Again, it's essential in mitosis, which we talked about in chapter 9, to make genetically identical cells. Because if all of our cells after fertilization are not genetically identical, they're all going to behave a little bit differently, and they won't really act in sync, and it'll be much more difficult to have big multicellular organisms doing complex tasks. So it's important after fertilization, during mitosis, to make genetically identical cells. But in meiosis, that's not so much the case. We want to increase our genetic diversity. And so in meiosis 1, that's going to happen through this crossing over phenomenon, where we're exchanging genetic material. So at the end of meiosis 1, it is different than mitosis because we create cells that are not genetically identical. So crossing over is this exchange of genetic material between what's called non-sister chromatids. So you don't have to know the definitions of sister chromatids and homologous chromosomes, but it just kind of helps with the discussion a little bit to show you where things are moving around. So here we have 2 pairs of chromosomes, and on each pair there's these sister chromatids. And then during crossing over, what's going to happen is these non-sister chromatids, so like the red and the blue, are going to trade genetic information, genetic material. So they're just going to physically trade these sequences of DNA. But these are all homologous. These are all like the same size and have the same genes. So when they share genetic material, it's just different sequences for the same gene. So it'd be like trading the eye color gene between 2 different chromosomes.
You just said we don't need no meaning.
Right, correct. Yeah. But I mean, if you wanted to just enrich your biology experience. That was making me so sad sometimes.
It's kind of, you gotta talk about it.
It's okay. So after crossing over,
Tell me about crossing.
happily. After crossing over, we've increased our genetic diversity, but we did so in a way that still makes all the cells functional, because we just traded specific genes. So imagine here, all of these sequences are encoding for the same types of proteins, just in slightly different ways. So it's okay to switch those between different chromosomes. And so after crossing over, we have each of these genes just on different chromosomes. So we've just increased our genetic diversity, but we still have like a functional instruction manual. So this is one of the ways that we can increase genetic diversity throughout meiosis leading into sexual reproduction. So meiosis 2, we're going to go from our 2 diploid cells and end up with our 4 haploid cells. So this was the final goal of meiosis, was to create these 4 genetically unique haploid cells. And so we did it partially through the crossing over and partially through this independent assortment that we talked a little bit about before, where each of these chromosomes can split independently of the other one. And so it can send different chromosomes to different haploid cells. And the more chromosomes you have, the more substantial this randomness from independent assortment can be. So after meiosis 2, we now have 4 haploid cells that can each serve as gametes. And they can all participate in fertilization with another organism to make a new diploid cell. So we have meiosis 1 and meiosis 2. At the end of meiosis 2, we've got our 4 gametes. Does anyone want to explain to me why meiosis 1 is different than mitosis? Or how they're similar?
Because it's one line and a whole lot.
Yeah, exactly. Yeah, so at the end of meiosis 1, we have cells that are not identical copies of each other. They're not genetically identical. But they are still diploid cells.
So in mitosis, how come like crossing over doesn't happen?
Great question. So let me make another drawing. I'm sure you all love my drawing skills. Did you say no?
I didn't say that.
No.
I just said no. I said no.
Oh yeah. It's not good, it's great. Yeah. It's kind of a dull color.
It's red.
Yeah, you're right.
Oh yeah. Wow. So mitosis during metaphase.
It's awesome. It's like Picasso.
When all of our chromosomes line up, they line up in the middle like this.
A true Monet.
What?
A true Monet.
Maybe I'll sign this up in my drawing.
What's a Picasso over there? What's the man that was on there?
So we have
The Afro. No. There you go. Like Bob Roy. Can't forget the man with the Afro. That's the way we painted it out.
well, yeah. Maybe I'll make this little.
See, you did that a lot. You know.
So during mitosis and metaphase, everything lines up in single file like this, whereas in meiosis, they undergo this synapsis process that we talked about earlier, where the 2 pairs will combine together like this. So we have non-sister chromatids that are touching each other in meiosis 1. Whereas in mitosis 2, you don't have the non-sister chromatids that are touching because they're all lined up single file. So the way what do you think would control the way they line up, the way their chromosomes move around?
The mitotic thingy.
The spin, yeah. So in this case, it'd be the meiotic spindle fibers versus the mitotic spindle fibers, but they're very similar. They're both these microtubule spindle fibers that are going to attach to all the chromosomes and move them around. So in meiosis, they're going to get moved around so that they can combine like this, so that non-sister chromatids can be touching each other, so that we can undergo this crossing over. And so the reason this happens is because organisms that do this and increase their genetic diversity tend to outcompete organisms that don't. So genetic diversity is kind of a behavior that gets encouraged because it increases like the fitness of the organisms that it produces and makes them more likely to survive. So animals or organisms that evolved to have meiotic spindle fibers that encourage this crossing over ended up being able to outcompete organisms that didn't, and that's why we have this trait. That said, organisms that would undergo this in mitosis would end up creating cells in a single organism that were genetically unique and not identical, and those would not do very well. Those would not thrive because they couldn't engage in like complex behavior. It'd just be like a total mess of different instruction manuals in the same organism. Is that a suitable answer?
Yes.
Great. So, summary, it's similar to mitosis, especially during interphase, And the individual steps of meiosis 1 and meiosis 2 are very similar to those of mitosis, but it's also very different because we're not producing identical cells in meiosis. So we end up producing these haploid cells, and for a male that'll be sperm, and for a female that'll be the egg, that can then combine to create genetically unique offspring.
For meiosis, there is a fifth era that goes back into the interphase.
Yeah.
What is that? There's no more.
Yeah, that's not really clear. That would have to be like fertilization.
Okay.
Well,
Unlike in mitosis,
yeah,
which
that's
is-
not a great. That's not a great.
Because mitosis goes on forever, but this has a beginning and an end.
Yeah. Yeah. Well, yeah, exactly. It was. Yeah, that's a good point. That arrow probably is not very clear. I think what they're trying to depict though is fertilization. I took this from a website that maybe had more context. I should have looked at that a little more closely. Or maybe this is coming from the germ cell that makes the haploid cells. But anyway, you're right that the 4 haploid cells up here don't go back into the cycle.
What I'm asking is there anything left of that cell? It divides and then divides and then there's nothing left. Right?
Yeah, exactly. It's just the 4 haploids. Yeah, yeah. So that's not really a helpful part of the diagram. All right, so there's 3 main contributions to genetic diversity. 1, crossing over, which we have this expert drawing that I still need to sign. I don't want to get it stolen.
Copyright.
So we have crossing over. This is a physical exchange of genetic material. So we're physically exchanging the sequence of DNA on different chromosomes to make genetically non-identical cells. Then we have this independent assortment, which means each chromosome can move independently of other chromosomes. And this allows us to create many different combinations from the same set of DNA in the original diploid cell. So these diploid cells up here have the same DNA within them, but as they make the haploid cells, they can each be different. So all of these are unique. We have all these unique haploid cells, even though they came from the same diploid cells. And the more chromosomes that you have, the more possible combinations of the genetic material you can have. So that's the second way we can increase our genetic diversity. The third way is just inheritance sexual reproduction. When 2 different organisms reproduce and share their genetic material, since they're non-identical, they're going to create new mixtures of DNA and create totally new offspring. So again, all of these ways of creating genetic diversity, they don't just like randomly mix up the DNA. They have to do it in a way that still preserves like the function of the cell and can still make a functional organism. But they want to increase the ability of that organism to survive in new environments. And so this is useful because the environments that organisms are constantly changing over time. And so by creating new genetic diversity with each generation, it makes the whole population more capable of surviving those changes. Does these 3 mechanisms make sense to everyone? Any questions about any of them? So when do each of these happen? At what point in the sexual reproduction or meiosis do each of these phases happen? When does crossing over happen? Prophase 1 and meiosis 1, yeah. So within meiosis 1, we have the phase prophase 1. And that's when we're going to have our cells line up, our chromosomes join together and share their genetic material. When does independent assortment happen? It kind of happens like all through meiosis 1 and meiosis 2. It just means that each of these chromosomes, as they're attached to the meiotic spindle, the spindle fibers all move independently of the other spindle fibers, which means that we can sort all of the chromosomes into different haploid cells. So independent assortment really happens all throughout meiosis 1 and 2, would be the best answer, I guess. And then sexual reproduction, that's going to happen during fertilization. So when we have 2 different haploid cells coming together to make a diploid cell. And just a comparison again. What? All right.
We were disappointed with the way you answered that.
Oh, what? Okay. So mitosis, again, identical cells, diploid cells. Meiosis 1, diploid cells, non-identical. Meiosis, oh, it should be meiosis 2. Sorry. Well, that was all meiosis. Meiosis 2 produces haploid cells that are all non-identical. Is that clear? So we have, where did my eraser go? Someone hid it from me. So mitosis identical. Oh my god. Sorry. You're gonna get this in the trash can from here?
That's close. Good try.
Just it's like.
Typical
Not talking, no.
meiosis,
No, nothing airborne.
non-identical
Air marker.
diploid. Oh, thank you. Sorry, I would have gotten non-identical haploid. So each of the phases is making different types of cells. All right, sorry, that was the last slide. All right, that's all I wanted to cover today.